Hello. This lecture for Chapter 5 goes through a lot of vocabulary that talks about some of the differences in patterns of inheritance that we see that are different from the phenotypic expressions we would expect from Mendelian genetics. So what I want you to understand is these genes are still inherited through the same process, laws of segregation, independent assortment, but the phenotypes are not the typical dominant recessive that we have been talking about. You won't be working very many problems from these, but you will have descriptions and you will want to be able to identify what kind of mode of expression is being demonstrated in a story problem. All right, so the first one we're going to talk about is incomplete versus codominance. And the key here, is okay, good. The key here is to look at, the heterozygotes. Okay, so we have big B, big B, big B, little b, little b, little b. With classic dominant recessive, anytime you have a dominant allele, you get the dominant phenotype. So these are the classic ratios we were talking about in class. 3:1 phenotype from a monohybrid cross, a 1:2:1 genotypic ratio. What you're going to see with incomplete and codominance is you still have the same genotypic ratio, but now each different genotype has a separate phenotype. So for instance, we have incomplete dominance where the homozygous, in this case is red. We have a different homozygous that is white, and the heterozygous, or the heterozygote, is pink. Okay, this is different than red and different than white. And the phenotype of the heterozygote is an intermediate. Okay, and it doesn't have to be exactly red plus white equals pink. Okay, but it is not red, it is not white, and as we'll see in a minute of codominance, it is not both red and white. So the inheritance of the alleles is the same, it's the expression of the phenotype that is different. And in this case, what's happening is the red allele, being only one copy, produces just less of the red pigment, and so you see this pink pigment. So the difference between incomplete and codominance is codominance means both phenotypes from the homozygous are expressed. So it's not a blend, it's not intermediate. Whoops. So in this case, you have a black chicken, a white chicken, and the codominant is checkered, which means both black and white on the feathers. Okay, they're not gray. Gray would be incomplete dominance. In fact, a roan horse is not pink, but it has both brown or chestnut and white hair being expressed. So it has this, looks like an intermediate phenotype, but in fact if you looked at the coat, it would have both brown and white. So incomplete dominance and codominance, you're always looking at the heterozygote. And you notice that we can represent them differently. So sometimes we excuse me, put a superscript that represents the two homozygous phenotypes. Sometimes we just use both capital letters because they're both dominant. Okay. Don't use big B little b and say that big B little b is checkered, because this indicates complete dominance. Another set of terms is incomplete penetrance and expressivity. And this is where the genotype does not produce the expected phenotype. And this is how a lot of genes are in organisms. And this is because there's genes that are interacting with each other. But complete penetrance means every organism that has a certain genotype expresses it identically. Or I should say expresses it. So complete penetrance, you have it, you express it. Incomplete penetrance means you have the same genotype, but not all organisms express the phenotype. And we can talk about penetrance as a percent. So in this case, since 3 out of 6 express, the penetrance would be 50%. Now, it can get even more complicated than that with expressivity, which is how much of the phenotype is expressed. Get rid of that so it doesn't get confusing. Okay, so that's shown down here, where here you have complete penetrance, narrow expressivity. So everybody that has it shows it, and they all look the same. You can have complete penetrance with broad expressivity, which means complete penetrance, everybody that has it shows it, but how they show it is different. And then, of course, you could have incomplete penetrance and narrow expressivity, which means not everybody that has it shows it, but when they show it, it looks the same. The expression is similar. Or incomplete penetrance and broad expressivity, which means not everybody that has it shows it, and those that show it have variable phenotypes. So you can imagine if you were a geneticist trying to understand inheritance, this would get super confusing, right? You could no longer follow nice Mendel's rules, because what you're seeing is not exactly what you'd expect with complete dominance, complete penetrance. So the example they use in your book is the allele for polydactyl, which you can have multiple fingers or toes, so more than the standard 5. And what this pedigree, and we're going to get into pedigrees soon, is showing you is that not everybody that has the allele, so this person's a heterozygote, sometimes they color that by half, not everybody that has it shows it. Okay, incomplete penetrance. Another example you may be more familiar with is like the BRCA1 and 2 mutations. So there is an increased risk with BRCA1, BRCA2 specific mutations to having breast and ovarian cancer. But it doesn't mean that everybody that has these mutations will get breast and ovarian cancer. So it has incomplete penetrance, and in this case also variable expressivity, depending on age and environment. The phenotype can be expressed differently. So the big take-home message from incomplete penetrance is just because you have a specific allele doesn't mean you're going to show it. Lethal alleles. We talked about this with achondroplasia. Lethal alleles generally mean death before birth or shortly after. Oops, death, birth. Okay. And the only way you know that there's a lethal allele is that when you do a standard cross, you don't get the expected phenotypic ratios. So you would expect for a heterozygous monohybrid cross a 3-to-1 ratio. And in this case, you're getting a 2-to-1 ratio. Okay, because one phenotype is lethal, and so you don't see those offspring. Okay. So this is the classic way to look at conditional probability. Okay. So we will talk about this in class again with achondroplasia. So in that case, if you have a big A genotype, most likely the child was not born, and if they are, they don't survive long. So the probability calculations are for the live births. Okay, probably not surprising. You can also have multiple alleles. So this idea that there's a big A or a little a combination really isn't how things are. Okay, you can have many different alleles for one gene. So the classic example is for blood type, and blood type would be your phenotype. And to determine blood type, we have 3 different alleles. We have the A allele, the B allele, and the kind of non-AB allele, which can result in type O, or can result in heterozygotes. Okay. The Punnett squares are exactly the same, but when you interpret the phenotype, you have different possible genotypes to equal the same phenotype. What's interesting about blood type is the reason we write it capital I superscript A, capital I superscript B, is these are in fact dominant and the little I is recessive. But what's also interesting is these guys, you notice they're both capitals. Together they are codominant. So if you have the A and B allele, you have blood type B. But if you have the A and the little I allele, you have blood type A. So you can be homozygous for A or heterozygous and still have the same blood type. There are also traits that are sex influenced. For example, baldness. And what sex influence means is these are autosomal genes, so they're not sex-linked. So they're on your chromosome pairs 1 through 22, usually influenced by hormones. And they're going to differ among the men and women. So you get different phenotypes for men and women. And in general, it again is the heterozygote that shows this difference. So in this case, baldness. If you are dominant for the bald, homozygous dominant, you will be bald in either sex. So men will lose their hair, women will have hair falling out, very thin hair. And if you're homozygous recessive, in both sexes you have normal hair. But in this sex influenced and heterozygote will be bald in males only, and females will be normal. So now you can kind of understand why baldness is more prominent in men than women. There are two possible ways for men to go bald. Oops. And only one way for females. Again, a lot of this is due to differences in hormone production. So it's influenced by the sex of the person. Sex limited means it's only expressed in one sex or the other. So there are some examples of feather production in chickens, for example. But one I didn't even think about, but it's kind of like a duh. Okay, milk production in females. Males do not produce milk. Females will produce milk upon pregnancy or the expression of certain hormones. Chickens lay eggs. Hens lay eggs. Roosters, the male, does not. So there are some common sex-limited traits that we probably just don't think about. And again, hormones are super important in the expression. Another kind of inheritance that looks different in the males and females is what we call cytoplasmic inheritance. So this is where you're getting DNA in the cytoplasm, not the nucleus. Not nuclear. So in the nucleus, you get DNA from your mom and your dad. But in the organelles, such as the mitochondria, and in plants, they have mitochondria and chloroplasts. These only show maternal inheritance. That means your mitochondria comes from your maternal lineage. Your mom, her mom, your great-grandma, not from your dad's side. This is because mitochondria and chloroplasts have their own DNA. This is also because upon fertilization, the egg contributes the mitochondria. So it contributes mitochondria, or mitochondrial DNA, and nuclear DNA. Excuse me. On the flip side, the sperm only contributes nuclear DNA. So, in fact, mitochondrial genes are used to trace someone's maternal inheritance. And you can trace that for males or females because everybody has mitochondria. But it doesn't tell you anything about your paternal side of the family. Another term used in genetics is anticipation. And in diseases with anticipation, as you look through a pedigree, okay, and so a pedigree is talking about phenotypes from generation to generation, what you start to see is you start to see earlier onset and earlier, oops, we're talking about age, or you may see more severe disease in each generation. So in this pedigree, you can start to see diagnosed at age 55, and they had cataracts. We'll talk about the repeats in a minute. Diagnosed at age 48, had grip myotonia, facial weakness. Right? Diagnosed at birth in this third generation. Facial weakness, neonatal hypotonia. So this is myotonic dystrophy, example of a disease that shows anticipation. Another one is Huntington disease, and what this pedigree is showing you is that the grandfather showed the disease at age 50. His grandchildren started showing it a little earlier. Their children started showing the disease even a little earlier. And the reason anticipation happens is many of these diseases are due to DNA mutations that can increase in accumulation. But it's not just an increase of mutations, it's an increase in this we call trinucleotide repeat sequence. So hopefully you remember somewhere back in your brain that every 3 DNA nucleotides encode for an amino acid. That's where the trinucleotide comes in, 3 nucleotides sequence. Okay. As DNA replication happens, as you start to accumulate these repeat regions, the DNA polymerase sometimes has a hard time keeping track of where it is, and it slips. And so it may inadvertently add more trinucleotide groups. Okay. And what that does to the protein is now it's adding more and more amino acids, and you're losing the function to a greater extent of the protein. So what this diagram over here, this table is trying to show you is that normally for people without Huntington, they have 10 to 26 repeats of this CAG nucleotide, trinucleotide. So they have 10 to 26 glutamines, and the protein functions fine. As you get to an increase of 27 to 41, we call it premutations. So there might be some issues in the protein function, and therefore in the onset of Huntington. And once you get to 36 to 121 of these repeat sequences, you become what we call affected, which means you will have and show signs of Huntington disease. So we don't have the repeat numbers on this chart, but what's most likely happening is they're accumulating more repeats and then passing that on, and their children are accumulating more repeats. And so you get an earlier and earlier onset of the disease. So that's anticipation. Couple more terms. Oh, I forgot I put this on. The other thing that you can see with anticipation at a molecular level is you can see an increase in the size of DNA. So this is a DNA gel, and what we can do is we can amplify a little region of the DNA and look at that gene sequence. And it's in fact hard to look at the specific gene sequence, because just like DNA polymerase in the body will kind of forget where it is, DNA polymerase that we use for gene sequencing will forget where it is. So what we do instead is we, if this is where the repeats are, we amplify outside, and we'll look on a gel at how big that piece of DNA is. And as the piece of DNA gets larger and larger, you can see the disease shows up earlier and earlier. So it's kind of a neat molecular technique. When we get to the molecular side of genetics, we'll talk about this a little more. There are also environmental effects. Sorry, I'm looking at my notes. Somewhere I put these pages. So for instance, temperature. So if you have a Siamese cat, you notice that their noses and paws and ears are darker. This enzyme works differently at cooler temperatures, which is your extremities of the body. I don't know if this actually works, but I thought it was an interesting way to demonstrate that if you put a cold pack on the little bunny, they would get a black patch. I haven't looked to see if that's true, but that's just to try to illustrate that the environment can affect gene expression. And this is kind of a, okay, makes a lot of sense. We see this. So what I want you to know is that the genotypes are identical, but the phenotypes are different based on the environment. So we see this with animals. Arctic fox in the winter and the summer, they change the color of their fur, right? So they're still the same fox, same genotype, but the phenotype can look different depending on the environment. Hydrangea plants, you can alter the soil pH and get different colored flowers. I think that's pretty cool. And a lot of this was done with twins, identical twins. So you know that some identical twins look very, very similar, but they could live or participate in a different environment that would affect their phenotype, right? So if you have an identical twin, just because they eat healthy and stay skinny, doesn't mean you're automatically going to be skinny, right? You've got to have the same environment to have those similar phenotypes. We also have something called continuous traits or quantitative traits. So here's where we were looking with Mendel. It was kind of a yes, no, tall, short, purple, white. But continuous traits means that you have a spectrum of phenotypes. So skin color is a great one, right? All you have to do is look around our classroom, and you would see a huge range of skin color. And this is due to multiple genes affecting the characteristic. So we'll put affecting the trait. Right? So it's not over here, one gene, one trait, but it's multiple genes and how they're expressed and what alleles you have at each of those different loci. So that's what polygenic traits mean. Many genes contributing to the trait. And this is how much of our genetics is, right? So eye color is not brown or blue. Brown and blue make green. Right? There's huge variations because there's many genes that control our eye color. And on top of that, you can have what we call multifactorial characteristics, which means not only many genes that affect that trait, but the environment has an effect too. So if we had had class a month or so ago, I was much more tan than I am now. My skin's starting to fade. I'm not getting my California sun like I did for a week in the summer. So not only is there a spectrum of traits for, say, phenotypes for skin color, but you could also have a whole nother based on sun exposure. Right, so multifactorial means there's many things going into this. You have multiple genes and you have an environmental influence. All right, one more term, pleiotropy. Pleiotropy to me is kind of the opposite of polygenic, and this means that one gene affects many traits. So, again, this is common for a lot of our traits. An interesting one is sickle cell. So, excuse me, if you have a sickle cell, not only does that gene affect the shape of your red blood cells, but the red blood cells break down easier, and they clump, and they accumulate, and that causes anemia and heart failure, brain damage, spleen damage, which can cause kidney failure and paralysis and mental issues. So one gene, and it's actually amazing, sickle cell, if you remember from general biology, if you went through it, sickle cell is one nucleotide change that makes one amino acid change. And that changes how the hemoglobin comes together, and people with sickle cell that are homozygous for it are very sick because that one mutation can affect multiple traits, affecting many different organs, right? Many different symptoms. So, what I want you to do from this is go through, write out your vocabulary. These are all in the learning objectives, or outcomes, sorry, learning outcomes. Learn them because in class we're going to start out with a whole bunch of clickers to see if you have learned your vocab. All right, that's it.